Hello everyone, my name is Adam Parker and welcome to my final project for Chem 394, Computational Chemistry and Biochemistry, proposed research of PolyQ mutations in the Huntingtin protein. The area I decided to focus on involves the Huntingtin protein and its relationship to Huntington's disease. For a little background, Huntington's disease is a neurodegenerative disorder where there is a progressive degeneration of neurons in the brain, usually leading to the loss of motor function control, cognitive loss, various psychiatric disorders, and eventually death. It is known that the Huntingtin protein is the cause of this disease, and more specifically, that the disease is caused by the expansion of the PolyQ or polyglutamine tract of the protein. Basically, it's a mutation of increased numbers of glutamine residues in this PolyQ tract, which leads to the disorder. However, scientists are not exactly sure how this mutation leads to the death of neurons. It is suspected that the Huntingtin protein is crucial to repairing damaged neurons, and in the mutated state, it can no longer fulfill this function, leading to the death of neurons. But there are also other ideas, and we'll get a little bit more into that here in just a little bit. So, a healthy Huntingtin protein contains a PolyQ tract of typically 9 to 35 glutamine residues, with the average usually being 17 to 20 residues. The mutation leading to Huntington's disease occurs when there are 36 or more glutamine residues in the PolyQ tract, and correlations of having more glutamine residues usually means that the onset of symptoms happens earlier in age. So I will be using computational methods to determine the effect varying numbers of glutamine residues in the PolyQ tract have on the energy and conformational properties of the overall Huntingtin protein. Neurodegenerative diseases are the focus of a lot of research these days due to how debilitating these diseases can be. The function of the Huntingtin protein is not well understood at this time. However, it is known that the Huntingtin protein regularly interacts with other types of proteins for normal functions in the body. What I've noticed is that current research in the Huntingtin protein is moving more towards computational methods to try and understand how conformational changes of the mutated protein can lead to altered interactions with these other proteins. It's also suggested by at least one research paper that these different protein type interactions with the mutated Huntington's protein may be what's leading to the different symptoms associated with Huntington's disease. More simulations are being done to investigate these interactions, as well as to explore the potential for inhibitors to prevent the mutated Huntingtin protein from even binding to these other proteins in an effort to lessen the severity of symptoms or even prevent the onset of symptoms of the disease altogether. Other computational research is being done on the interactions between healthy Huntingtin proteins and mutated Huntingtin proteins, suggesting that this may also be leading to the severity of the disease. My first resource is current therapeutic molecules and targets in neurodegenerative diseases based on in silico drug design. The authors of this paper believe that computational methods are the most efficient, moral, and cost-effective way to come up with new treatments for neurodegenerative diseases. There are ongoing molecular docking studies being used to determine the interactions of the mutated Huntingtin protein with its known associate proteins, such as protein kinase C, casein kinase substrate, FIP2, GRB2-like protein 3, and postsynaptic density 95. The focus of their research is in silico drug design, which is using computer simulations and modeling software to design multi-target inhibitors to prevent the interactions of mutated Huntingtin proteins with these other named proteins. The second resource I used was molecular mechanisms of heterogeneous oligomerization of Huntingtin proteins. The researchers of this paper focused on the interactions of mutant Huntingtin proteins and non-mutated Huntingtin proteins. To do this, they used discrete molecular dynamics simulations which follow protocols designed for potential energy calculations based on molecular collisions. This allowed them to create more realistic scenarios of molecular interactions to understand the aggregation of Huntingtin protein molecules. They determined that in order for Huntington's disease to occur, aggregation of mutated proteins need to form in the brain to interfere with normal functions. They used different lengths of PolyQ tracts to determine how this affects mutated Huntingtin and non-mutated Huntingtin protein interactions. Their simulations revealed that a single mutated Huntingtin protein can aggregate up to 2 wild-type Huntingtin proteins. They then correlated these findings by using fluorescence lifetime imaging microscopy, fluorescence resonance energy transfer, to actually observe what they had found in their simulations. So that's where I picked up for my project, to test varying numbers of glutamine residues in the PolyQ tract to see what the energy and conformational changes are, and eventually to see how those different conformations affect how the Huntingtin protein interacts with those other proteins that we talked about earlier. So my plan of proposed steps was to first load a computational model of the Huntingtin protein containing a normal starting number of glutamine residues in the PolyQ. And then I plan to make several mutations of varying numbers of additional glutamine residues to the PolyQ, and then run molecular dynamic simulations of those Huntingtin protein models, and then evaluate the results to determine the energy and conformational differences of the Huntingtin protein with the varying numbers of glutamine residues in the PolyQ. And then the data may be helpful in understanding how the Huntingtin protein functions in different conformations, and the data may also explain why 36 or more glutamine residues leads to Huntington's disease, and why there is such a large range of varying lengths of the PolyQ, from 9 to 35 glutamine residues are okay, and how those actually interact with the other proteins, even in normal conditions. And then to see how the mutated Huntingtin protein with 36 or more glutamine residues changes from those to, now non-functioning with those other proteins, and leads to Huntington's disease and the individual symptoms associated with it. So what I did first was to actually obtain a PDB of the Huntingtin protein from the rcsb.org protein database site, and this protein contained 14 glutamine residues in the PolyQ tract. I used the mulfacture tool in VMD to then mutate the protein by adding glutamine residues of varying numbers for a total of 15 glutamine residues, then 19 glutamine residues, 24, 26, 35, and then ultimately 36 residues in the PolyQ tract. I then used QwikMD to convert the PDB files to PSF using the automatic PSF builder for all variations of the protein, and I attempted to load the protein molecules into Avogadro to generate the Gaussian input files and run geometry optimization. However, every time I tried to do this, Avogadro crashed, and I was unable to run the geometry optimization. But anyway, I still went ahead and prepared the non-optimized proteins in QwikMD for molecular dynamic simulations in VMD, and I attempted to run those simulations in VMD. However, after a few hours of each simulation, it kept timing out before it was complete. So here's a demonstration of the steps that I took. First, I obtained a PDB of the Huntingtin protein, which contained 14 glutamine residues in the PolyQ tract, from the rcsb.org PDB site. The ID for this protein is 3 India Oscar 6 for your reference. And here's an image of the protein with the polyglutamine tract highlighted in green on the left, and then on the right you can see it more zoomed in with the rest of the protein in that purple color and the PolyQ Q tract highlighted in green. I rendered these images using VMD. The next thing I did was to use the mole factor tool in VMD, which allows you to add and subtract atoms and molecules. So I used this tool to add varying numbers of glutamine residues to the PolyQ tract. And you can see on the far left, I started by adding one glutamine residue, which is highlighted in orange, compared to the rest of the PolyQ tract in green, for a total of 15 glutamines in the PolyQ tract. Then in the middle image, I added more glutamine residues for a total of 19. And then on the far right, you can see that there's a total of 26 glutamine residues, and also in all of these images you can see the newly added residues are in orange compared to the previously existing green glutamine residues from the original PDB file. So I continue to add glutamine residues to the Huntingtin protein using the mole factor tool in VMD. And the more I used it, the more proficient I got and was able to add this many glutamine residues. So if you look at the image on the left, there's a total of 35 glutamine residues in the PolyQ tract, which is the most you can have while still having a normal, healthy, functioning Huntingtin protein. On the right, I added one more glutamine residue to the PolyQ tract for a total of 36 residues. And at this point is when the Huntingtin protein becomes mutated and can no longer function correctly, which leads to Huntington's disease. I thought this type of mutation was very interesting and fun to do, and different from the rest of the mutations that we learn about in class, running through the tutorials. So I think adding new residues was a unique way to mutate a molecule. At this point, I attempted to load the protein molecules into Avogadro to generate the Gaussian input files and run geometry optimization and single point energy output. However, this is where I ran into a problem. And every time I tried to upload the protein molecules into Avogadro, the program would crash. And I was unable to determine the cause or fix this issue. So I'm not sure if it was a problem with my input files, the PDB files or PSF files that I created, or the mutations that I did by adding the glutamine residues, but it still crashed when I tried to upload the original PDB file containing the non-manipulated Huntingtin protein. So I was unable to move forward with geometry optimization. I wanted to continue with my project and hopefully obtain some data from this. So I pressed forward and loaded the non-optimized protein molecules into QwikMD and prepared files for simulations using VMD. I was successful with that part and then attempted to run the simulations. However, they kept timing out after a few hours of running through the simulation. And I'm not sure if this was because there's a set amount of time that you can use the ASU servers, or if it was a connection issue between my computer and the servers, or if it was just an issue with my computer or with the actual files themselves. And anyway, I tried running the simulation just on my computer, and I don't think I have the computing power to do it because it also failed for each molecule I attempted. So that leads us to where to go from here. And I propose that the next steps be to optimize all the Huntingtin proteins with the varying numbers of glutamine residues in the PolyQ tracts, and then run geometry optimization and single point energy output for each variant of the protein molecule. That way we can compare the optimized energy values and conformational variations of each version of the protein. And the next step to take it even further would be to compare these conformational variations of the Huntingtin protein with the other protein types that are associated with it for normal functions in the body, and to see how these interactions are altered, because it is such a big variation in allowable glutamine residues in the PolyQ tract from 9 to 35. So there's bound to be conformational changes within that range, and to see how these interact with the associate proteins, and then to move on to see how the mutated Huntingtin protein conformations interact with these proteins, leading to Huntington's disease. So I think there's a lot of interesting places that this project could have gone, and maybe one day will go. To end this presentation, here are the articles that were referenced earlier. Thank you.